Hello, amazing people of BMS community and everybody else as well. I'm really excited to be meeting you through this BlueSciCon 2026. A quick intro about myself. As my name suggests in the display, myself, Madhumitha Srinivasan. I'm an incoming master's student in University Paris-Saclay, and this summer I got this amazing experience of being the YSP's RA and under Dr. Tomasz Zatkowski, and I was working along with Team Pioneers. So let's get into the actual project that I was working on. So the topic is PRIONOSONS, Prion-like domains of transposable elements as models of transient evolutionary scaffolds. That's a lot of terms, but by the end of this project, you'll be able to understand what exactly I'm trying to talk about in this entire topic. So before we get into the actual concept, so every time I always personally like a storyline when the character goes through a huge character development, especially when a bad guy turns out to be the good guy at the end of the entire storyline. And something like this happened in molecular biology as well. So here I am talking about it. So in when you see towards the late sorry. Just a second.
No worries. Is the image not showing up? It's getting cut off?
Yeah. It's fine. Yeah, so as I was saying, towards the late 20th century, there seemed to be a discovery of a specific new element called as prions. This was specifically associated with transmissible spongiform encephalopathies, a type of neurological disorders. And these were found coined the term prions, which is a short form of proteinaceous infectious particle, and it also received the Nobel Prize for it. So for a long time, prions were seen as this really bad pathological character, but as research progressed, we were able to identify towards the earliest 21st century that prions might be associated with other beneficial factors or beneficial traits as well. And that is when the concept of prion biology began to be explored further. And what's so unique about prions is that their biology itself is slightly beyond what we understand about life, gender life, as we see in today's modern biology itself. So whether you're a biology student or a non-biology student, everybody must have come across this concept of central dogma. It's basically just the flow of the information itself. And we all know it goes from DNA to RNA to proteins, and it gains functions and everything. But what happens in prion biology is that it is quite unique. It slightly extends the central dogma, one can say. So what happens in prion biology is that initially the prions are encoded by genes, and they create what is called as a native state, but they can undergo spontaneous misfoldings and form spontaneous aggregation, a different kind of conformation. And this conformation can further stack up with each other to form myeloid formations. But at the same time, these kind of structures can also have an influence on other proteins. And in a way, the protein conformation itself is becoming a template that is moving from one to another. And it does not involve the information that has to be encoded by the genes or the genetic element. So that gives us a question. Could prion biology be an extension of what we understand of central dogma itself? So with all these interesting concepts about prions, so how exactly do we study these prions is the next topic that we have to think about. Well, there are two major ways in which we can study prions. And first one is through these regions called as prion-like domains. So these prion-like domains are very unique to these prion proteins, and these are specific intrinsically disordered domains, and they are very rich in specific amino acids, glutamine and asparagine, and they can be identified among sequences, and they are involved in phenotypic switching as well. The limitation, though, is that when we study a prions with the help of sequence analysis, we understand that prion behavior itself is not only prone to the sequence, it is something that happens in the next level as well. So we cannot just rely on sequence data and confirm it as to be a prion, and we can only call it as prion-like domains and prion-like behavior, something like that. Well, then we need to think of a more stable and approach, and that would include the experimental analysis. And the most common one is the gold standard is done by Reed Wigner, his East Prime criteria. However, the scope of this YSP project is going to rely mainly on the prime-like domains because we'll be using bioinformatic tools and analysis. So we had to pick a pill, so had to pick a pill, and that's going to be prime-like domains as the main character in our story. Well, next we have to talk about the second main character, which is transposable elements. So far, we know that genes are much stable and stationary and everything. Well, maybe sometimes it's a little bit jiggly. There are things called as transposable elements, and these are kind of a genetic disruptors. So these are mobile segments of the genetic gene itself, which can displace itself from its original position to another position. So it also had a very interesting storyline similar to that of prions. Earlier, it was associated as something called as a junk DNA, and when it was first discovered by Barbara McClintock, even though it's been 50 years, it's still a huge marvel of a mysterious marvel of molecular biology. Generally, it's been classified into two classes based on the replication mechanisms that is involved. Well, the similar story that I was talking about with respect to prions and transposable is that transposable elements was also considered as hallmarks of neurological disorders initially, and they were seen as the bad guy. But later on, it was found that it also produces siRNA molecules to sometimes suppress themselves and make sure it doesn't cause too much trouble to the genes. And it also involves in genomic plasticity to increase some molecular adaptation as well, which talks about the next question. Could there be some kind of an association between these two main characters, which seem to have a very similar storyline? Well, we did think about the associations as well. When you talk about life, it is mainly divided into three major domains, which is archaea, eukarya, and bacteria. Well, prion-like domains occur in all three domains of life itself. That is one major interesting fact that we know. And the second one is these prime-like domains. They are present in although they are present in all the three domains of life. They are very uneven, and they have a very sporadic distribution across different protein lineages itself. Sometimes they are present in sometimes in even closely related proteins they are not present, and that raises another question. However, even if they are very unevenly distributed, these prime-like domains were present in proteins associated with DNA transposition across all the three domains. And especially to go a step further, it was experimentally found that transposable elements seem to have a region that has a prion-like property involved in it, which gives us way to the next thing, which is the gap in literature. So when we talk about adaptation does require a heritable mechanism, but it needs to be somewhat reversible at the same time. And in the early stages of adaptation itself. And both prions and TE seems to do this. So prions are doing it through phenotypic switching of DNA repair and recombination proteins without directly going to the gene and changing this basic element, the blueprint of the species itself. At the same time, T is also doing that in a way. It is creating genomic plasticity by inserting itself, activating itself in different conditions and contexts. And both of them are supporting this kind of a mechanism in some way or the other. So the missing piece at the end of this entire story is, can prime-like domains and T act together as temporary solutions for this phenotypic variation error? Which brings me to my hypothesis, which is PRLTs represent a temporary evolutionary scaffolds whose adaptive role is eventually replaced by a more stable genetic or structural solution. Well, another story time, but this one is the actual conceptual framework that I'm proposing here. So let us assume a situation where there is a requirement for some kind of a change. So what could happen here that these domains, the prime-like domains, could be recruited by a specific protein in just like how in a new project, an intern could be recruited in order to help them in the earlier stages of establishing the protein project itself. And these newly acquired domains can be recruited through different evolutionary mechanisms, probably through transposable element activities or through viral acquisition, etc. And once these proteins have acquired the prion-like domains, they might have a more advantageous effect over this evolutionary period itself. So they can gain an enhanced capacity for change in the form of probably reversible molecular interactions or biomolecular condensate formations or oligomerizations, etc. And this kind of, even though it's transient, this kind of a reversible mechanism can help natural selection to go over different choices and choose the one that is preferably better. Then leads to the next step, which would be functional stabilization. So after a long period over when this adaptation process is going on, the protein itself could favor a more stable motive or a stable conservation mechanism or a regulation mechanism. And this prion-like domains become somewhat like that is not necessary anymore. So the protein starts becoming rather independent of this prion-like domains activity itself. Which means at the end of all of this, the prion-like domains become somewhat redundant, and to have a very aggregation-prone and unstable signature, sequence signature within the proteome, it's not very, it is not very adaptable, or it is not very good for the protein to survive itself over the long run, because of which some kind of erosion mechanisms has to happen. And this could probably happen by truncation or amino acid substitution, etc. So when you take a step back, if you look at this entire mechanism, how would it look in the actual perspective is that if we look at the ancestral sequences versus the descendant sequences, the prion-like domain signature or the presence of prion-like domains must be weaker in the current or modern descendant proteins compared to that of its ancestral proteins. So that way, some kind of erosion is happening in the prion-like domains itself within the protein. Which brings us to the final question: how do we exactly analyze this? How do we study this? And those were my objectives in this YSP project as well. So first, we can analyze spatial distribution. So that basically we will try to identify the prion-like domain signature across viral, retroelement, and host-associated proteins. And then we need to identify signature that is not just 0 or 1, but some kind of a gradual content. That is, it must be strong prion-like domain signature, weak prion-like domain signature, and some kind of an erosion pattern, etc. And finally, as we saw in the earlier slide, the ancestral sequence relatively must have a stronger prion-like domain signature compared to that of the descendant proteins, that is the modern proteins that we see right now. How did I go about that? Well, in order to identify the Apache distribution, I was able to collect different data sets from different databases, mainly of 3 categories: retroelements, which is a class of transposable element, endogenous retroviruses, and retroviruses. Why these 3? Because they have been identified to have very similar mechanisms and very similar structure, etc. So comparative genomic analysis was done using MAFT. And if anybody wants to go over the in-depth over all the different sequences that was acquired, I'll be happy to explain it afterwards as well. And the second one was to identify different patterns of prion-like domain signature, that is strong, weak, etc. So this was done using a very interesting tool called BLACK. BLACK is this bioinformatic tool that it basically gives you a spectrum of scores of the prion-like domain signature itself. So higher the core score is basically stronger prion-like domain signature, lower it is, lower the signature. And finally was the ancestor versus descendant sequence analysis. So this is done using IQ3. And again, the plaque was run both on ancestor sequences as well as the descendant sequences. And these ancestor sequences are basically a prediction based on what descendant sequences that we have in our databases. So when we compiled all the data, the results, basically transposable elements, that is Ti1, Ti2, Ti3, Ti4 elements of GAG proteins, they seem to show a slight difference in the ancestral sequence prion-like domain signature to that of the descendants, whereas endogenous retroviruses showed even more difference in the ancestral versus current descendant proteins. And HIV was the that is a retrovirus, it was the most interesting result out of it. There was a huge gap between the ancestral sequence prion-like domain signature and the descendant prion-like domain signature as well. So MGE is the final run that has been going on, but this is to extend from gap proteins to other proteins as well. And it is an additional next step kind of thing. So to summarize, at the end of this project, I was able to produce a conceptual theory or a framework for the associated priority in evolution and formulate a methodology and do a comparative analysis as well. Overall, the prion-like domain signature showed from strong prion-like domain signature to apparent loss through the ancestral sequence to descendant sequences. The next step would basically be to check if there is some kind of functional stabilization also happening alongside the erosion and check for enrichment, etc. So if everything is proven right, well, I review the PRIONOSONS, which is temporary assistance of evolution itself. Yes, so with that, I thank my entire team pioneers and BMS community and my mentor as well for supporting me through the entire project. So, any questions? I'm ready to take that as well. Thank you.